day everyone, and also to you Miss Moira Abismo. Today we discuss about effect of increased soil pH and pH buffering capacity induced by crop residue biocars on available CD contents in acidic paddy soil. Before we start, let me introduce myself. First, I am Jason G. Talian, and also would like to introduce my members: Angeline Fernandez, Reza Canido, Ginny Carros Galela, and Iverson Cartala. Research address the significant environmental issue of cadmium CD contaminations in acidic paddy soil, which is poses as threat to human health through rice consumptions. The authors highlight that soil acidifications not only leads to aluminum phytotoxicity and nutrient deficiency, but also enhance CD availability, increasing its uptake by rice plants and ultimately affecting humans' health through dietary intake. This study aims to investigate the potential of crop residue biocars in mitigating CD contamination by exploring their impact on soil pH and pH buffering capacity (pH-BC).
Objective. First, evaluate the impact of biochar amendments on soil pH and pH-BC after laboratory incubation. Second, analyze the relationship between biochar-induced changes in soil pH and pH-BC on the available CD content. Third, assess the effectiveness of biochars in inhibiting the decline of soil pH during drainage, a key factor in CD activation. Fourth, investigate the correlation between soil pH-BC and the change in available CD content during submerging draining cycles. Fifth, determine the effect of biochars on CD absorption by rice plants, thereby reducing the potential risk of the soil CD to humans.
Materials and methods. First, soil election. Search acidic paddy soil with varying origins were selected for next time. Second, biochar preparation. Biochars were produced from seeded sunflower seed, peanut straw, and corn straw using pyrolysis. Third, laboratory incubation. The biochar were incorporated into the paddy soils at different concentrations and incubated for 30 days at 25 degrees Celsius. Fourth, soil analysis. The pH-BC and available CD content of the soils were measured before and after incubation. Fifth, submerging or draining alternation. Soil samples were subjected to alternating submerging and draining cycles to stimulate technical paddy field condition. Sixth, rice root experiment. Rice plants were grown in biochar amendment soils to assess the impact on CD uptake.
Results. The addition of crop residue biochar significantly increased both soil pH and pH-BC in all 6 paddy soils, while reducing the available CD content. The effectiveness of the biochars in decreasing CD content was in the order of SSPBC, PSBC, or CSBC, correlating with the increase in soil pH induced by each biochar. The biochars effectively inhibited the decline in soil pH during drainage, compared to control soils due to their enhanced pH buffering capacity. A strong negative correlation was observed between the change in available CD content and soil pH-BC during submerging draining cycles suggesting that higher pH-BC leads to lower CD availability. Rice plant grown in biochar-amended soil show significantly reduced CD uptake, confirming the biocar's ability to decrease the potential risk of soil CD to humans.
The study findings strongly support the potential of crop residue bicars as effective soil amendments for mitigating CD contamination in acidic padding soils. The increase in soil pH and pH-BC induced by the biocars directly influences the availability of CD-reducing its uptake by rice plants and ultimately decreasing human health risk. The authors emphasize the importance of the biocar's ability to stabilize soil pH during drainage, which is crucial for preventing CD activation and minimizing its potential release into the soil solution. The strong negative correlation between soil pH-BC and available CD content further supports the biocar's role in the immobilizing CD and reducing its bioavailability.
In this image, impact of soil pH and pH-BC on cadmium CD. Low soil pH and pH-BC. High CD availability leads to increased CD uptake by rice plants, resulting in higher health rates for herbivores and humans. In high soil pH and pH-BC, biochar addition increases soil pH and pH-BC, reducing CD availability, uptake, and associated health risks. And role of biochar: rice straw biochar improves soil conditions by increasing pH and pH-BC, thereby lowering CD bioavailability. In CD uptake by rice varies during flooded and dry conditions but is significantly reduced in biochar amendment soil. In health risk pathways, high risk, high CD levels in rice lead to greater risks for herbivores consuming rice straw and humans consuming rice. And low risk, reduced CD levels from biochar amendment lower health risks for herbivores and humans. Food chain contamination. The schematic highlights the flow of CD contamination from soil to rice, then to herbivores and humans, emphasizing biochar as a mitigation strategy.